In prokaryotes, DNA replication begins when initiator proteins bind to the origin of replication, a small region of DNA containing a specific sequence of bases, creating a complex. This complex helps to initially separate the DNA. Then, the enzyme DNA helicase binds to it and continues to unwind the DNA by breaking the hydrogen bonds between the complementary strands. The newly opened areas are stabilized by single-stranded DNA binding proteins. Each one can now serve as a template for the synthesis of a new strand of DNA. The unwinding and synthesis proceeds in both directions from the origin, creating two replication forks. In front of the forks, topoisomerase enzymes bind to the DNA and reduce torsional strain as the molecule unwinds. Once the strands are separated, another enzyme, primase, synthesizes an RNA primer, a short stretch of RNA complementary to the DNA sequence. The primer provides a place for the enzyme DNA polymerase to add nucleotides complementary to the DNA sequence, creating a new DNA strand in a process called elongation. DNA polymerase synthesizes DNA in the 5' to 3' direction of the molecule, so the synthesis of this strand, the leading strand, proceeds continuously. The other strand, the lagging strand, has the opposite orientation. Consequently, DNA is synthesized in short pieces called Okazaki fragments, elongated from additional RNA primers, backwards from the overall direction of movement of the replication fork. The RNA primers are then excised by enzymes such as RNase, replaced with DNA, and the DNA fragments are joined together by the enzyme DNA ligase, creating a continuous strand. DNA replication proceeds around the entire molecule, resulting in two circular DNA molecules. This is considered a semi-conservative process because each molecule contains one old strand and one new strand. Most prokaryotic factors utilized during replication have equivalents that play similar roles in eukaryotic DNA duplication. This process initiates at an origin of replication to which a recognition complex binds. Helicase is then attracted to the site and separates the strands of DNA, generating a bubble with two forks. Primase also arrives and generates RNA primers, which, as helicase moves, DNA polymerase elongates with new DNA. As in prokaryotes, the newly formed leading strand grows continuously, following the replication fork. Conversely, the lagging strand is manufactured in small Okazaki fragments, traveling opposite the fork. Due to multiple factors, the DNA template used to generate the leading strand in one half of this structure creates the lagging strand in the other. Interestingly, various origins of replication exist on a linear eukaryotic chromosome, and replication terminates when their associated spheres coalesce. Primers are then eliminated via enzymes like RNase and swapped for DNA. Afterwards, DNA ligase attaches any segments. However, when the end primer disappears from the lagging strand, the space remains empty, and there's an uncopied stretch of DNA template abutting it. To combat this, an enzyme called telomerase affixes to the overhanging region and elongates it with a non-coding DNA sequence. Primase and DNA polymerase act upon this extended region, creating a telomere cap that protects against loss of coding DNA from the lagging strand during multiple replications. Thus, eukaryotic DNA replication ends with two DNA molecules, each with a parental and newly synthesized strand, numerous origins of replication, and telomeres.